Hello everybody, I'm Silas, and welcome to Psilocybology. Let's expand our mind. This is a series where we talk about the research on psilocybin, the psychedelic prodrug found in magic mushrooms. We'll be breaking down and discussing the scientific literature, one study at a time, from past to present. If you'd like to support the show, please make sure to rate, review, and subscribe to the podcast. Today's episode is on the article titled Clinical Syndromes and Biochemical Alterations Following Mescaline, Lysergic Acid Diethylamide, Psilocybin, and a Combination of the Three Psychotomimetic Drugs, published in 1964. Now yes, you heard that right. As stated in the title, this study did administer a combination of mescaline, LSD, and psilocybin, and compared the results to the administration of each drug separately. The researchers were, quote, curious to see if combining these three similar drugs would alter their overall clinical and biochemical effects. Sounds interesting. Unfortunately, the findings are pretty lackluster. The other thing I want to note about the aim of the study is that the goal was also to replicate some of what these researchers have found in prior experiments. The first author of this paper, Leo Hollister, was an assistant clinical professor of medicine at Stanford. He was also an author on the manuscripts we discussed on episode 6 and episode 16 of the podcast. In episode 16 in particular, the researchers examined a set of biochemical outcomes after the administration of mescaline, LSD, and psilocybin. In this study, we'll be able to see if a few of those findings were replicated. I'll provide a brief but more detailed recap of what was previously found once we get to that section of the results later in the episode. Getting straight into the methods, a total of 24 participants were ultimately allowed to participate in this study. The researchers noted that any history of emotional problems, psychotherapy, or even prior psychedelic drug use were exclusionary criteria. Part of the rationale for this is that the study is not designed to be therapeutic, and often when a study is not designed with a treatment focus, then researchers prefer to use healthy participants. I'm sure, however, these authors were also buying into the fear that was being unjustly spread about these drugs. I'm basing that primarily on their problematic usage of the term psychotomimetic rather than psychedelic in the paper title. As we discussed previously, psychotomimetic essentially describes drugs that produce a psychotic-like experience, and was promoted in part after an unsubstantiated rumor was spread that a female killed herself after taking LSD, due to a psychotic episode induced by the drug. The problem here is that the evidence actually shows time and time again, and we'll see it again in this study, that although psychedelics alter our perceptions of reality, they do not sever our connection to it, particularly in the sense that individuals taking psychedelics remain oriented to who they are, in that they know their name, their age, etc. They are aware that any hallucinations they experience are caused by the drug, and although their understanding of time and place might be altered, if you ask someone tripping questions like what year it is or what country they're in, they're able to answer them accurately. This is all why in episode 19 we discussed the argument made for ditching the term psychotomimetic for psychedelic. Anyway, once they got their 24 participants, which were unfortunately all males, they then assigned each participant a sequence of drug trials. I say unfortunately all males because, as we also see in respect to race and other demographic factors, often research is conducted using one specific group and is then erroneously overgeneralized to the entire population without properly investigating how the effects may vary between groups of individuals. Now, in many cases, it's possible there is no difference. Humans are, after all, far more similar than we are different, considering we share about 99.5% of our genes. That said, one reason having a representative sample is important is that even if our genes are similar, people are shaped by vastly different upbringings and social conditioning that is based on sex, race, gender, sexual orientation, income, etc. This, I'd argue, is particularly important for examining psychedelics, given how strongly mindset can influence psychedelic experiences. Sorry, that was a bit of a tangent. Getting back into the methods. This was a within-subject design, so each participant received each of the 4 drug conditions, separated by 1 week. The researchers used a Latin square order to determine the sequence of drugs for each of the participants. A Latin square order just means that they made it so each of the 24 participants received the drugs in a unique order. Since there are 4 conditions, there are 24 possible combinations, hence why they needed 24 participants. The 4 conditions were as follows: 1.5 micrograms of LSD per kilogram of body weight, 5 milligrams of mescaline per kilogram of body weight, and 0.225 milligrams of psilocybin per kilogram of body weight. And then finally, for the combined treatment, the researchers administered 1/3 the single dose of each drug simultaneously. All drugs were administered in white capsules, without either the subject or experimenter knowing which treatment was being given during any one session. Moving on to the measures. There were 3 self-report assessments used in this study. The first was a 75-item questionnaire designed by the researchers of this paper, which they say was built on the 45-item questionnaire they used in their previous publications. The questionnaire essentially just asks about the presence of somatic symptoms, such as nausea, dizziness, etc., perceptual symptoms, such as hallucinations, and psychic effects, such as changes in mood. Response options ranged on a 4-point scale, from no, meaning not present at all, to whole lot. This questionnaire was administered 1.5 hours after drug administration, and again 6 hours after administration. The second self-report assessment was called the Clyde Mood Scale. This test consisted of 133 cards, each with an adjective describing a feeling or state of mind, such as sluggish, kind, cheerful, fearful, confused, nervous, rebellious, etc. Participants were asked to sort the 133 cards into 4 piles, each pile representing how much they currently feel that emotional state, from not at all to extremely. Depending on how participants sorted the cards, researchers can group certain terms and calculate subscales for different mood factors. Specifically, the researchers in this study chose to look at the scores for friendly, energetic, clear thinking, aggressive, jittery, and depressed. Participants did this assessment before the drugs were administered, and again 1 hour, 3 hours, and 6 hours post-administration. The third and final self-report assessment was called the Hoffer-Osmond Diagnostic Test, which consists of 145 true-false statements asking primarily about perception as well as cognitive and emotional disturbances. One aim of the questionnaire was to distinguish between patients with schizophrenia from other psychiatric patients and healthy controls. The items can be analyzed such that you end up with a score measuring perceptual abnormalities, paranoid ideation, and depression. Some of the statements include, people's faces seem to change in size as I watch them, most people hate me, and life seems entirely hopeless. I want to note, though, that some of the statements just seem rather pointless and even dated, like, a dress is like a glove because they belong to a woman, rather than because they are articles of clothing. I feel like the way one answers that has much more to do with one's conception of gender roles, rather than whether they are experiencing disturbances in thought. Another weird one, other people's cigarette smoke smells strange, like a gas. Which, if we're talking states of matter, then smoke is in fact a gas. I get what they were going for, but still, this assessment seems somewhat poorly designed to me. Some of the items do make sense, and appear to have face validity, but too many don't for me to give the assessment as a whole too much credence. In addition to the self-report measures, the researchers also took urine and blood samples prior to and during the experiment. I found it interesting and funny that they specified that water was given if urine flow did not soon begin, when they were attempting to get urine samples specifically just prior to drug administration, and specifically 2 hours post-administration. Time for the results. Starting with the 75-item symptom questionnaire. The main result is that there were no significant differences in the number of positive questionnaire responses between the 4 conditions. Of the items endorsed, the researchers also noted that, to their surprise, very few of the items were perceptual or psychic, meaning that few participants reported symptoms like visual hallucinations, depersonalization, distortion of body image, etc. The respondents primarily endorsed the somatic symptoms, so nausea, dizziness, drowsiness. The doses given were decent in magnitude, so I think part of the problem might be in the questionnaire itself. They never actually provide the questionnaire, so I can't tell for sure how these were worded. But just going off the examples they provide, I see 3 potential problems. First, there seems to be more negative symptoms given as examples, such as difficulty with attention, difficulty with verbal expression, poor memory, paranoid thoughts. My problem with this is in part that I think the positive or negative valence may vary a lot among psychedelic experiences, as well as one's own perspective. Assessing broader changes in attention, verbal expression, without assigning a value, might produce better results. The second problem, although along similar lines, is that some neutral symptoms are framed as negative, such as disturbed depth perception or distorted body image, rather than just altered depth perception or altered body image. The third and most important problem is that depending on how they worded the question, participants might not know what the researchers were asking them. For example, if you just asked, "Are you experiencing synesthesia?" how many people do you think will actually be able to answer that accurately without any further explanation as to what synesthesia is? People will know whether they feel nauseous, but they may have no idea whether input from one sensory modality is producing a response from another, let alone that experience is a transient synesthesia. It's an absurd question. Given the issues with the questionnaire, I wouldn't draw too many conclusions from the data. Although they didn't find any differences between the groups in this study, keep in mind most of the responses were of somatic symptoms. So if anything at all, it just means that these 4 conditions didn't produce drastically different effects when it came to things like dizziness, fatigue, nausea, etc. The next set of results deals with the Clyde Mood Scale. These results, I think, are rather telling. They found that across all 4 conditions, participants scored higher on the depressed, jittery, and aggressive subscales after drug administration. Clear thinking, on the other hand, decreased across all 4, and the friendly and energetic subscales decreased after mescaline and remained the same after the other 3 drug conditions. As the researchers themselves point out, this pattern of results, where the negative symptoms seem to increase and the positive seem to decrease, is at odds with many other accounts of the effects of these drugs. To me, my first thought is, what was the set and setting like? Unlike the study we covered last week, which described in detail the lengths those researchers went to into providing a comfortable and supportive environment, and the subsequently positive experiences that it engendered, these researchers don't mention the study environment at all. And my concern is that they didn't provide a setting conducive to positive psychedelic experiences. The last set of data from the self-assessment is from the Hoffer-Osmond diagnostic test. The key finding that the researchers present in the paper is that the statements least often answered positively were those relating to genuine psychotic symptoms, including things like delusions, which are unshakable beliefs in something false and impossible, despite evidence to the contrary, and paranoid thoughts, like the example we provided earlier of, "most people hate me." They also noted that, similar to the 75-item questionnaire, far fewer items concerning perceptual changes were endorsed in this test than they expected. Finally, we have the biochemical measures from the blood and urine samples. Specifically, the researchers examined blood and urine phosphorus, circulating eosinophils, circulating free fatty acids, and blood glucose. Now, I don't usually break up a study over multiple episodes. However, I want to wait to talk about the free fatty acids and blood glucose till next week. The reason is because next week the paper we'll be talking about is by the same author and is entirely focused on free fatty acids. So for today, let's just focus on the other key outcomes, starting with blood and urine phosphorus. Back in episode 16, we discussed how phosphorus is a really key component to a lot of biological processes, and some prior work had shown a decrease in the amount of phosphorus excreted in the urine after LSD administration. Given its importance, there are potential consequences to too much or too little serum phosphorus. However, mild to moderate deficiency has been found to be generally well tolerated. What the researchers found in the study from episode 16 was that participants given mescaline, LSD, or psilocybin all exhibited a decrease in the amount of phosphorus excreted in the urine. However, there were no changes in serum levels of phosphorus. This indicated that the reduced amount being excreted is not resulting in excess levels building up in the blood, or a result of a deficiency in the amount of phosphorus being produced. In the current study, the researchers again found a decrease in the amount of phosphorus excreted in the urine. However, they also found a slight increase in the amount of serum phosphorus as well. That said, the increase of serum phosphorus is very low, and the values are still well within the normal range of 2.5 to 4.5 milligrams per deciliter for adults. The increase seen from psilocybin, for example, went from 3.21 milligrams per deciliter to 3.52 milligrams per deciliter. This to me actually seems like a great opportunity to illustrate the differences between a statistically significant finding and a clinically meaningful one. In this case, they found that the increase from 3.21 to 3.52 was statistically significant, meaning that the increase was unlikely caused by chance alone. However, since it is still well within the normal range, I'd argue it doesn't carry a lot of clinical significance. If instead the change was from 3.21 to 5.21, then it'd clearly carry both statistical and clinical significance, since the increase pushes it outside of the normal range for adults. Always try to keep that distinction in mind, because sometimes there will be findings that are statistically different, but the difference is still so small that it isn't very meaningful. And last but not least, let's look at eosinophils. Again, as a recap from episode 16, we discussed how eosinophils are a type of white blood cell that fight parasites and certain infections. Although beneficial, high levels of eosinophils are associated with tissue damage and asthma. This is a lot like how inflammation is caused by your body protecting itself from infection and illness, and while not having an inflammatory response would be a big problem, having chronic inflammation can also end up causing a lot of harm as well. So in this case, no or not enough eosinophils would be bad, but too many are also potentially problematic. In both episode 16 and episode 6, the researchers found that psychedelics lowered eosinophil count. In the current study, they didn't find any statistically significant reductions of eosinophils. However, they did note a trend that eosinophils seemingly declined 2 hours after the 4 drug conditions. It's called a trend just because it didn't reach the threshold needed to be significant. The only reason I even bring up that there was a trend in the same direction is to say that I wouldn't use the absence of an association in this study to discredit the presence of an association in the prior studies. It's possible that an increased sample or perhaps a stronger dose would have produced significant findings. So overall, I can't say we can take away too much from this study, particularly the self-report, although it does possibly further highlight the importance of set and setting in producing positive experiences. That said, there weren't at least any drastic differences seen in the combined category compared to each drug separately. I also still think this was an interesting study to cover, and had some good educational takeaways. And as I mentioned, next week we'll focus specifically on free fatty acids, so stay tuned for that. Well, that's it for today's episode. As always, I really hope you enjoyed the show. If you have any questions, feedback, or ideas for the podcast, please let me know. You can find out all the ways to reach me on the website, psilocybology.org, where you can also find the full transcript for each episode if interested. Thank you all for listening, and I'll talk to you next time.